In the year 2000, a falling tree killed a female Pyrenean Ibex named Celia in the mountains between Spain and France. When her heartbeat stopped, her entire species went officially extinct. 3 years later, scientists successfully used preserved skin cells to clone her. This was the first time a vanished animal was pulled back. But the newborn Ibex lived for only a few minutes before her lungs failed. The experiment proved that even when extinction is undone, the results are incredibly fragile. You can see the unique physical texture of the gastric brooding frog, an Australian species that swallowed its own eggs and burped up live young. When a team tried to bring it back using frozen cells, the embryos grew for only a few days. Traditional cloning strictly requires perfectly preserved living cells, a resource that is incredibly rare. Because animals that have been dead for centuries, like the woolly mammoth or the dodo, have no living cells left, traditional cloning is a biological dead end for them. Scientists have largely moved away from the pursuit of perfect genetic clones. Instead, they are using modern gene editing tools like CRISPR to write a different kind of comeback. This is the proxy method. You start with the genetic code of a closely related living cousin and edit the DNA until it matches the traits of the extinct animal. The goal isn't to create a museum replica, it is to engineer a functional, living animal built to reclaim its specific role in the wild. Before you can edit any DNA, you have to read the original instruction manual for the species you want to bring back. This image shows the distinct stripes and predatory build of the thylacine, also known as the Tasmanian tiger. Though it has been gone since 1936, its biology is no longer a mystery. Researchers recently took a century-old museum specimen and successfully sequenced its entire genome, mapping every single letter of its genetic code. Recovering that data means the complete biological instruction book for the thylacine is now open and readable on a computer screen. With the blueprint in hand, scientists need a living canvas. They use the DNA of a closely related modern cousin to begin making active edits. This schematic shows a gray wolf genome. To recreate the dire wolf, scientists edited 20 markers across 14 target genes. These edits force the return of the ancient predator's traits, triggering the development of a broader head, heavier frame, and thick coat. The same logic applies to the Asian elephant. Because its DNA is nearly identical to the woolly mammoth, its physical structure serves as the perfect canvas for mammoth traits. To prove the concept, scientists recently edited 7 mammoth genes into laboratory mice. The result was woolly mice that grew long, wavy, golden coats. The success of these tests confirms that we can effectively force modern genomes to express extinct, ancient traits. Once the DNA is edited, the final hurdle is growth. How do you turn a petri dish of edited cells into a breathing animal? For the thylacine, being a marsupial provides a shortcut. Because the young are born the size of a jelly bean and grow in a pouch, a small relative can handle the pregnancy without needing a massive surrogate mother. Birds are much more difficult. This is the passenger pigeon, a bird that vanished in 1914. Because birds develop inside hard-shelled eggs, they cannot be cloned or gestated using standard mammalian methods. The workaround involves editing specialized pigeon cells and injecting them into surrogate chickens. These chickens act as living bioreactors, eventually laying eggs that will hatch into proxy chicks. Solving these incubation problems is what allows us to move a genetic sequence off a screen and into the real world. We have to be clear about what these animals are. They are living proxies, engineered to survive today rather than serve as perfect genetic duplicates of the past. This is the rowan antelope. It is currently the biological baseline for the bluebuck, a species extinct for 2 centuries. Because the rowan can act as both a genetic donor and a surrogate, this project is moving faster than almost any other. The timeline is no longer theoretical. Proxy direwolves are already alive and growing on a private preserve. Mammoth calves are targeted for late 2028, and passenger pigeons are expected between 2029 and 2032. Extinction is no longer a permanent end. Within our lifetimes, these genetic sequences will become the living giants walking our wild spaces once again.